Sheba, can you start your presentation?
Sure, just a minute.
Mm.
Can you see my screen?
Yes.
So, good evening, everyone. My name is Sheba, and today my topic of presentation in the journal meeting is titled Rapid Recombinant Protein Production in Mammalian Cells. A brief overview of what I'll be delving in. Sorry, just a minute. I'll first define what a recombinant protein is, and then discuss various expression vector systems that are used in the production of the recombinant protein. I'll also discuss transient gene expression and the limitations that exist within the system, and then further go and discuss about the cell culture and how we can optimize the condition in the media for optimal production of the recombinant proteins. Also, I'll discuss gene delivery methods that are currently in use, with the main focus that will be polyethylenamine as the gene delivery method. And lastly, transient transfection in recombinant protein production of particularly recombinant interferon gamma using PEI, and lastly conclude on the overview of this discussion. So I think most of us have heard of recombinant proteins, but a brief explanation. It can be defined as a polypeptide that is usually produced by cloning genes through recombinant DNA techniques. And generally, a heterologous expression system is used, meaning that the gene of interest is inserted into a foreign host that naturally does not produce the protein or encode for it. So the process usually begins with isolating the gene of interest, that is the DNA fragments. And once it's identified, it's isolated and then characterized. And once it's optimized, it can be inserted into a vector through recombinant DNA technology. So here's a brief schematic of how it's done. Usually, the target gene that you want to maybe express in a system is selected. And after the gene of interest is established, you choose an expression vector whereby you clone the target gene into the expression vector, after which the cloning and the sequence is confirmed, it's transfected into a host cell. And for the recombinant protein production, there's different systems that are currently in use, but commonly the use of mammalian cell lines like the H293 T cells and CHO cells are common because they have various advantages regarding the post-translation modifications and production of soluble proteins. Insect cell lines are also a popular method of recombinant protein production, also because they have post-translational modifications and can be produced in high cell density, and also soluble proteins are able to be produced. But the most commonly used in especially large-scale bioreactor or companies in industries is the E. coli. This is because of its ease to scale up, rapid expression, and low cost. But the disadvantage it has is that it has no post-translation modification, has inclusion bodies, and molecular weight limitation, which may affect downstream purification procedures. We can also use yeast. It also has various advantages and disadvantages. So after transfection into the host cell, transient gene expression, the crude protein produced is purified using different methods, and hence you can be able to obtain the particular recombinant protein of interest. Currently, there is growing interest in the rapid production of recombinant proteins, particularly cultured in animal cells, because they have diverse uses in both medicine and also health research. So transient gene expression differs greatly from stable transfection because you're only expressing the gene temporarily in a cell, and hence the genome is not entirely changed, but it's just expressed for a brief time through the transportation of gene into the cell nucleus and no incorporation into the cell genome. Currently, the two most widely used systems in recombinant protein production involve the use of mammalian cell line or insect cell lines, whereby transient gene expression is done in mammalian cells, and the baculovirus expression vector is used in infection in production of recombinant protein in insect cells. So what are the major differences between the two? the two differ in various characteristics. One includes the production duration, whereas the baculovirus expression system has a production duration of 2 to 5 days. The transient gene expression can be longer, up to 2 weeks. In terms of host cell cytotoxicity, baculovirus is a system that is cytolytic to insect cells, but for the transient gene expression, there's no cytolytic effects to mammalian cells. And also in terms of construction of the vector, whereas the construction for the baculovirus expression system vector can take time, for the transient gene expression in mammalian cell line, it's faster and takes a shorter time through the cloning that is employed at the moment. And also the biggest difference between the plasmid DNA requirement is that in transient gene expression, a high amount of DNA is usually required compared to the baculovirus expression system. For both, one can be able to scale, but it's higher in terms of cost for transient gene expression. And the protein yield for both are moderate to high. But the common thing between the two is that in both there's post-translational modifications that is supported, and hence they can be both ideal and hence can be optimized to be used in recombinant protein production. So while both systems can support correct post-translational modification, only mammalian cells produce complex glycosylation patterns needed for therapeutic proteins and therapeutic applications. So it's the most desired and best method for production of recombinant proteins. So my major focus for the discussion today will be on transient gene expression in mammalian host cells, such as the 293T and the CHO cells. And for host cells, particularly 293T and CHO cells, usually, especially in large bioreactors, they need to grow to a high cell density of greater than 5 to 10 to the power 6 cells per ml in suspension culture, and also have a transfection efficiency of greater than 50% for success in transfection and also the production of the recombinant proteins. Also, another condition that is vital is that serum-free growth is preferred, especially for proteins that are secreted, be it the intracellular protein or transmembrane, or even the just the secreted protein to the media, because serum can sometimes affect the downstream purification. So the less amount of serum that's there, the better the purity of the recombinant protein will be. Additionally, for these reasons, both of these two cell lines are preferred because they have these advantages of growing to high cell density and also ability to grow in a quick manner in serum-free media, and also have correct folded and post-translational processed modifications. Hence, they are best in terms of meeting the criteria for recombinant protein production of various systems that are currently applied. So the maximum level of protein accumulation usually is observed on day 2 to 3 post-transfection, specifically for transmembrane and intracellular proteins. And on the other hand, for secreted proteins, ideally it could be 3 to 7 days, and sometimes it can also range till 2 weeks maximum, that one can obtain the highest level of DNA after doing recombinant protein production procedure. So what are the limitations of transient gene expression? Whereas we found success in production of various recombinant proteins, there's various challenges that exist in this system. One includes the scaling up of transient gene expression is very costly and challenging, as one needs to do pre-transfection and ensure that the cells are constantly passaged and to an optimum volume to ensure that production is optimum. Additionally, whereas DNA uptake is not a limiting factor, sometimes the transgene transcription reaches a level where it plateaus, and hence sometimes the amount of DNA you need to be taken up is not possible. Additionally, researchers have found that in transient gene expression, most intracellular plasmid DNA can remain intact, but sometimes exist in linear or nicked form. So sometimes it's unclear to know which form is transcriptionally active, but researchers have been working on this. And lastly, lot-to-lot variation. We know there's different types of commercial media available for production of recombinant protein, but sometimes the fact that the quality differs can affect the variability in terms of how the cells remain viable during the production process, or the maximum cell density that can be achieved, transfection efficiency, protein yield, and other factors. So these are the various challenges that exist that need to be solved as one optimizes how to produce recombinant protein in suspension cultures. So as I've mentioned before, these two cell lines are the most commonly preferred in terms of production of recombinant proteins because they have various advantages. And if you focus on 293T human embryonic kidney cells, in terms of their genetic background, they have this gene which ensures transcription occurs optimally, hence conferring it and the advantage of being used in the recombinant protein production system. Additionally, the simian virus 40 large T antigen enables episomal replication of plasmid to boost mRNA expression, hence the preference for this type of production of recombinant protein. It has high transfection efficiency. Also, it can be highly adaptable to suspension culture because the increase in the episomal plasmid replication and the gene copy numbers enhances the yield of the protein and hence the versatility in used in both recombinant systems for various therapeutic proteins. CHO cell line is also the same, and there's various variations that exist, and they're widely used in the bioproduction system in recombinant protein production. So that was a brief introduction on the reason why 293T and CHO cells are preferred and how transient gene expression is done through the recombinant DNA technology. So as I mentioned before, when culturing cells in suspension culture for recombinant protein production, the most important aspect is considering the media components and how they affect the yield of whatever gene of interest you want to produce through this platform. So media components and how they're regulated is an important aspect, and I'll briefly discuss how scientists currently optimize the media to ensure that proper production of recombinant protein in various systems. Suspension cultivation of the HK293T and CHO cells are usually performed in serum-free media that support densities of up to 5 to 10.6 cells per ml in large-scale bioreactors, but this can be adjusted depending on the scale you're producing. Maybe in the lab, there's also an optimal amount of cell density that's required, but preferably serum-free media is preferred to help in optimal production. Additionally, when culturing or optimizing cells in for recombinant protein production, herpes is usually added that acts to maintain pH stability as it's a deuterionic buffer with a pH of 7.3 at this temperature. And usually it's added in addition, or it's commonly used together with sodium bicarbonate at concentrations ranging from 10 millimolar to 25 millimolar to ensure the effective buffering. Hence, the cells are able to thrive within that particular period of time through this condition. Another component that is usually adjusted or optimized during the recombinant protein production is the addition of 1,X-glutamax, which is usually a stabilized form of glutamine that is added to prevent degradation and ammonia buildup, which can be toxic to cells. The main advantage it confers is that it ensures nutrient production by providing the essential nutrients required for the growth of cells. And another component that is vital is the coliform P188. Usually when cells clump, maybe from aggregates that are produced or foreign, rather the DNA that is produced during cell lysis, the clumps that form can hinder nutrient and oxygen diffusion, and this means that there's depletion of nutrients and oxygen, meaning that the cell viability is not at its optimum and the protein expression is repressed. So by addition of the coliform P188 and anti-clumping agent, this can be are deterred and ensure protein expression occurs in an optimal manner. Lastly, valproic acid is another component of the media that can be added during the transfection process in a suspension culture. It's a histone deacetylase inhibitor, which removes acetyl groups to ensure the recombinant protein, particularly in the cell line, is optimized in terms of the mRNA expression in a protein and cell independent manner. Usually, the valproic acid, as it's a histone deacetylase inhibitor, removes acetyl groups, and hence the chromatin structure of the cells that are being cultured are more open, and hence the DNA is more exposed to transcription to ensure high levels of mRNA is produced, and hence enhancing protein expression of the recombinant protein of interest. In terms of choosing the expression vector that will be used for the transient gene expression, diverse expression vectors are used, but particularly one commonly used vector is the pcDNA 3.1 because it has various components that range from the CMV, which is a strong constitutive promoter and enhancer, to enhance the transient expression in both CHO cells and 293 T cell line. Additionally, this expression vector is preferred because it has certain ampicillin-resistant gene, neomycin-resistant gene, multiple cloning sites to allow ease for insertion of the gene of interest during the cloning process, as you introduce the gene of interest for cloning to produce the transient gene expression system. In terms of the gene delivery method, the most cost-efficient methods available right now are the use of calcium phosphate or the cationic polymers, commonly the polyethylenamine. And especially this one is preferred as it's a non-viral gene delivery method that is not only cost-effective and scalable. So the linear 25 kilodaltons polyethylenamine is used at 1 milligram per ml of concentration, and it has the greatest advantage as one can use it without requiring addition of serum in comparison to calcium phosphate that requires addition of serum prior to transfection. And before, it was a bit complicated in terms of optimizing the transfection using PEI, but now it's been established that the direct addition of to the cultured DNA followed by PEI is possible, whether you're transfecting at low or high cell density. Hence, it's a very direct method of transient gene expression as long as you optimize the condition of the cell and the density before you start transfection. So the use of PEI as a transfection reagent usually works through the positively charged PEI ensnaring the negatively charged DNA to form this complex, the DNA PEI complex that now binds to the cell surface. Then it's taken up through the endosomal vesicle and released to the plasmid, to the cytoplasm after osmotic soiling. So usually the final recombinant protein yield is affected by variables even after the transfection, and hence the need for understanding how the cell culture condition affect the DNA yield at the end of the experiment and even after the downstream purification processes. So going forward, I'll discuss previous research and how they've optimized the experiment to enable us better understand how the DNA PEI ratio, the serum concentration, or the temperature of the incubator affects the production of the recombinant protein of interest that they were producing. The research paper that I'll discuss was focused on production of recombinant interferon gamma protein using the PEI, and they wanted us to understand how certain conditions could be adjusted to optimize the production of this recombinant protein in an optimal manner. So a brief explanation of the interferon gamma. It's a secreted protein, a cytokine, belonging to the type 2 interferon family, which has roles in both innate and cellular immune response. And usually it's produced under inflammatory condition by T lymphocytes or natural killer cells, and hence it's an important protein, especially in cancer immunotherapy and other types of disease research. So I'll discuss different aspects of how they optimized their system to ensure the optimal production of this recombinant interferon gamma protein. And these are the four major things I'll delve on. So the first thing they wanted to analyze was the toxicity of PEI. And to do this, the researchers cultured CHO cells with a DNA pair complex with this plasmid at a predefined ratio of 1:8. And it was established that a PI concentration of greater than 30 microgram resulted into more than 50% of cell death when the trypan blue exclusion dye assay was done. However, PI at less or equal to 10 microgram per 5 ml showed low toxicity. So with this experiment, they were able to postulate that PI fixed at 10 microgram per 5 ml could possibly be optimal for ensuring proper transfection without causing toxicity to cells and causing unregulated death of cells. In also determining how efficient the CHO cells are at transfection, the researchers transfected CHO with this vector, the Pmax green fluorescent P vector, at the DNA pair ratio of 1:8, and observed the cells at 12, 24, and 48 hours, and then observed it under microscopy. So figure A1 was under the bright field microscopy showing successful transfection, and figure A2, they were able to observe green fluorescence, confirming that the transfection was successful, and this was an overlap of the two images. So the histogram that was generated from this experiment that was done in triplicate showed that 50% of CHO cells were green fluorescent positive at 48 hours post-transfection using the linear PEI, hence suggesting that transient transfection of CHO cells could be achieved at relatively high levels using this particular linear PEI based on these experiments that were done. So after confirming the amount of PEI that's optimal and the use of CHO cells as being successful in ensuring transfection efficiency, the researchers wanted to confirm what exact ratio is permissible to ensure optimal yield of the DNA of interest in recombinant system, particularly the recombinant interferon gamma protein. And so to do this, they cultured the cells, the CHO cells at different ratios of DNA and PI, as shown here, from 0 to 10 to around 1 to 12. And accordingly, the yield of the recombinant interferon gamma protein was shown to increase as the DNA PI ratio was increased, as you can see here, 1 to 2, 1 to 4, 1 to 6, and the optimal was again confirmed to be 1 to 8. So after any increase after 1 to 8 was shown to be detrimental, as there was decrease in the DNA yield, and hence the optimal ratio of the DNA to PEI complex was determined to be optimally 1.8, and anything above that was shown to be not healthy for the production of quality DNA as shown in this experiment. Lastly, they wanted to optimize the culture conditions, and to do this, they used two hypothermic temperatures, that is 32 degrees and 37 degrees, which is the normal human temperature. Accordingly, as shown here, they observed the cells at cell growth at 2 days, 4 days, 7 days, and 14 days to establish the amount of DNA that was produced. And optimally, 7-day post-transfection was shown to have the highest yield of around 326.09 microgram per ml of the recombinant interferon gamma protein at 32 degrees. But at 37 degrees, even with a different concentration of the serum, the DNA yield was found to be lower. that is 2.69 microgram per ml at 37, and hence the difference in temperature was found to be very critical, as there was a 121-fold increase of interferon gamma yield at 32 degrees Celsius, which is really low compared to that was produced at 37 degrees Celsius. The researchers also wanted to establish how the serum concentration affected the yield of the quantity of the DNA. And to do this, they optimized the system to do two different temperatures, again 32 and 37 degrees, and used 10% fetal calf serum and 0.5% fetal calf serum at both conditions. Accordingly, it was shown that it did not particularly affect the yield of the DNA. Optimizing or reducing the fetal calf serum concentration is only a critical step because it improved cost effectiveness, especially in the downstream purification procedure. But otherwise, in terms of the yield of the DNA, no big difference was found to be present post-transfection. So to conclude, the researchers in these two papers wanted to establish which conditions are optimal for the recombinant protein production, especially for large-scale bioreactors, and also how the system can be improved by choosing the correct cell line to optimize the growth accordingly. The culture temperature is something that is very significant, especially in the recombinant protein yield, and optimal results were shown at 32 degrees Celsius. Additionally, serum concentration in the media had no impact in the recombinant interferon gamma production, but it's good to consider reducing it because of the downstream purification and to avoid complications in that. Additionally, the best DNA-PEI ratio was found to be 1:8. With this, anything above this will cause too much toxicity and death to cells, so optimizing it in this ratio was found to be the best option. And lastly, maximum recombinant interferon gamma yield can be achieved using 10 microgram PI in 5 ml using this media at 32 degrees for 7 days. So we've been currently trying to produce different types of recombinant protein, and we've used these two papers as a guide, and hopefully it's going to be successful, and this marks the end of my presentation. Thank you for listening.
질문. Actually, you reviewed the expression system in CHO cell, right?
Yes.
And now we are trying to express protein 293 T cell, right?
Yes, we're trying with 293.
Is there a little, it can be a little, the difference is between the CHO and 293 T cell. There's no comment about that in the paper, in review paper? No? Okay.
I think there is, but I can't particularly recall.
Actually, I remember in case of CHO cell expression system, when the company use serum-free media, they recommend some additional nutrition for production of recombinant protein, right?
Right.
I remember what kind of protein source we use. Maybe they recommend this kind of this kind of I cannot remember. It's similar the component used for bacterial culture. Trypsinized. Okay. So for the serum-free, the production of recombinant protein mammalian system, when we produce the recombinant protein, we need to add additional nutrition, right?
Yes.
It definitely helps to increase the production of recombinant protein.
True.
But maybe we need to consider when we adding the additional nutrition, we need to consider the pH, because usually the additional nutrition usually decrease the pH of medium, and also the osmolarity. Too much difference in osmolarity inhibit the cell growth and protein production. So 2 points when we need to consider about that.
Oh, Sheba, I have a very simple question. For my understanding, for if you're using HEK293 cell, that means you have to do something else than you're doing the E. coli production. As you know, even you using the E. coli, you can produce the interferon gamma for sure. There's a protocol. And what is the reason for using for 293, HEK293 for the interferon gamma? Why? Because you're not, I don't think you're just increasing production and everything, because it's post-translation modification is the key point, right? Why?
I think using the mammalian cell line offers more advantage because the recombinant protein that is produced I think it still goes around to the post-translational modification that ensures its stability and solubility and that it maintains its bioactive form after the downstream purification process. So I think this is the major advantage of using the mammalian expression system. I believe so.
For my understanding,
you showed us the table comparing the E. coli, yeast, and baculovirus system, and then mammalian system. This one.
Oh, this baculovirus.
Oh, no. Previous one. Another table.
This one, maybe.
Oh, right. Okay. Usually, the recombinant protein for medical use need a correct 3D structure, right?
Right.
For making the correct 3D structure, the mammalian cells use a variety kind of tools are used inside the cell, right? Making 3D structure is very, very difficult, right? That's why 2024 Nobel Prize conferred to the group AI-based 3D structure, the prediction system. We call that AlphaFold 2. So 3D structural determination is not simple. Making correct 3D structure is also very, very complex process. Especially the secreted protein in mammalian cell use ER, Golgi, secretory vesicle system. During the polypeptide chain passing through ER, Golgi, and then secretory vesicle, in all the steps, the polypeptide modified for making correct conformation, 3D structure, and also in terms of glycosylation. Glycosylation process is also not simple. From from the ER, they advanced until the Golgi and trans-Golgi network so very, very complex.
Right.
So when we express the recombinant protein for human use, expressed in E. coli, basically no 3D structure, no 3D, no 3D. We cannot expect the correct 3D structure. So usually we purified the recombinant protein, making the insoluble pellet inside the cell. We call that inclusion body. So when we making the recombinant protein by using the E. coli system, cheapest one, right?
Right.
At first we lyse the E. coli and then collect the inclusion body, insoluble inclusion body. And then with the inclusion body, we start the step-by-step denaturation steps. In some the denaturation condition, maybe the recombinant protein having the similar 3D structure, but never the we cannot expect the PTM, post-translational modification in E. coli. So the little more advanced version of recombinant protein production is yeast production system. But in yeast, some kind of from yeast to the insect baculovirus, larva cell, insect cell, we can expect some kind of the post-translational modification, for example, some glycosylation.
Right.
But never similar with the human glycosylation. Actually, in human cells, depending on cell type, glycosylation patterns are very different. So the field of glycosylation is very, very complex. But maybe the CHO and 293 T cell system is better in glycosylation, definitely than E. coli and yeast.
Right.
So, for my understanding, using HEK293 cell for not interferon gamma making antibody, and then I know there's a two way to making antibody. You ever heard the protein motif or epitope from antibody? Have you ever heard of
Yes.
that? So, nope. There's anyway, there's a two way to making the antibody by using HK293. One is making the increased affinity of antigen by the 3D structure, and another is making more binding sites on antibody for antigen. So that's what they're targeting for 3D structure, making antibody. And then even the interferon gamma is a similar thing in the site for the cytokine, try to find which site or which epitope is the better way. So there's a two-way. There are lots of scientists targeting for HEK293 cell for 3D structure. One, as I said, one is increase affinity, another is increase binding site. Oh, another thing, why E. coli is not affordable for 3D structure? Why? What kind DNA does using for E. coli for the making protein? What kind DNA? Which DNA? Which kind DNA?
Um, I I I honestly maybe I don't know.
Normally using the plasmid DNA.
What?
Plasmid DNA for the E.
Plus.
coli.
Actually, the for mammalian cell expression, we also use a plasmid, right?
Right.
So the gene of interest, the gene we want to express the cloned as a seed form of cDNA, complementary DNA, cDNA. But actually cDNA is only the connection of a coding region sequence, right? Coding region sequence. Only cDNA also only contains CDC, coding region sequence, as DNA, double-stranded DNA. So basically, in terms of expression of the mammalian expression, the mRNA produced by the plasmid is different from the original, never, right? Different, because we artificially connect only the coding region sequence. Especially in the mammalian cell contain the long, contain intron, right? So basically in pcDNA, the upstream region of the coding region cDNA sequence contain the intron-like structure, the DNA portion, intervening sequence inserted in the cloning vector, which help to increase the protein mRNA expression. And also when we choose the expression system, we need to consider which promoter we use, right?
Right.
For each systems, their own promoter should be used. For example, for E. coli, we need to add the viral the bacterial promoter. We usually use T3 and T7 promoter. T3 and T7 promoter is from the bacteriophage the DNA, because the virus only can infect to the bacteria. We call that bacteriophage. So bacteriophage have a very, very potent promoter, which can be used as in the E. coli. So T3, T7 promoter is used for bacterial expression. But in case of yeast and when we use the larva cells, there were a unique promoter needed, right? It's like the CMV promoter is from the CMV virus, cytomegalic inclusion virus, CMV virus. The CMV is a very potent promoter, viral promoter, which can be which works well in the mammalian cell, especially 293 T cell, not CHO cell. So for CHO cell expression, we need to consider another good promoter, which can be used in the CHO cell. And several more difference. For the mammalian expression, we also need to add a poly-A tail sequence, poly-A signal. Do you know what is the poly-A signal? Most of mammalian mRNA is after the transcription, primary transcription, need to modify in three ways. One is 5' capping, and then the second one is mRNA splicing. So the during the splicing process, the intron regions are mostly deleted, and then coding region connected. And last one is at 5' end, usually poly-A tails are added. So which poly-A tail definitely stabilize mRNA, mammalian expressing mRNA. So in pcDNA vector, the poly-A signal locates the five prime terminal end of the CDC cloning region. So little expression system little different. Definitely different for expression system. So by this system, what are you doing now? Can you explain about that?
I'm trying to produce certain recombinant proteins. In the past, we've tried with TNF. The secretary
TNF.
believe it was successful. Now we're trying.
So it was successful?
But the expression wasn't very high in Kumasi, but definitely for the secretory TNF, we got a positive result.
Really?
Yes.
Are you sure? I cannot remember. Okay. You tried the 2NF in the 293T?
No, we tried in CHO cell.
CHO cell.
But now it's the gasdamin E. We're trying to clone it through the transgene expression in pcDNA with the C-terminal tag polyhistidine. So
why you use the gasdamin E with a C-terminal polyhistidine? Why?
The histag protein, the system we're using for purification employs the affinity chromatography immobilized system. With
what kind of affinity purification system can be used with for the histidine tag?
The immobilized metal affinity chromatography. Because it has the nickel luciferase column that has high affinity to the histag protein.
Okay.
So through this system, we can be able to purify it during the downstream purification together, gasdamin with the histag protein.
That is very old system. So nowadays, I found on some paper different system nowadays you use. For example, streptavidin binding protein, peptide. Instead of histidine, some people add at C terminally add streptavidin, streptavidin binding peptide streptavidin SPP motif at C terminal, and then by the streptavidin agarose bead, we can easily more specifically purify the recombinant protein. I saw at the very high level paper, but his tag is most popular. Okay, I hope you in this week you can get enough gastronomy. E.
Yes,
okay?
I also believe so.
Proceed to the next step. Okay? Okay. 쉐바가 클로닝 실험을 되게 잘해요.
아, 그래요?
클로닝. 바이러스도 만들었어요. 지구상에 없는 바이러스.
나중에
Do you
생산공장
remember
만들어야지.
recombinant VSV? VSV 베스큘로스토마이타이스 바이러스를 지놈에다가 인플루엔자 베스큘로스토마이타이스 바이러스가 그 바이러스의 인플루엔자 바이러스의 그 코트 프로테인들을 두 개, right? 헤마글루틴 유람인데 코팅되는 리컴비넌트 바이러스를 만들었어요. 그거 클로닝 하려고 그때 what was size exact size of a recombinant genome BSP genome?
It was almost 12,
12, 12? Not 12, 20, 20, 20. Not 12, 20. 20 지놈 만들었어요. 근데 익스프레션 레벨이 너무 낮아가지고 때려쳤는데
음,
어쨌든 만들었어요. 그게 쉬운 건 아니라는 거 저도 잘 알고 있어요. 우리 학생들은 질문 하나 한번 한 학생만 질문. 칸, 미스터 칸, please question to Sheba. 미스터 칸,
I don't have a question. Sorry.
예이.
I'm sorry.
우리 또 한규리, 한규리 학생, 한규리
네,
질문 하나만 해줘요.
모르겠어요.
우리 이소림 학생, 우리 질문 하나.
없습니다.
네, 우리 김도 김독 김 김도경
김도경.
학생, 김도경 학생 질문 하나만 해주세요. 김도경
어,
학생, 네?
네, 저도 딱히 질문 없습니다.
이게 요즘에 이게 돈 되는 테크닉이래죠. 이게
맞아요.
이거 이렇게 리컴비넌트 이 단백질 만드는 시스템 중에 제일 비싼 게 오늘 쉐바가 발표한 내용인데요. 요거로 신약 그 뭐지? 그 대체 뭐지? 그걸 무슨 약이라고 그러더라? 타겟티드 세러피, 항암 종양
네네네.
치료에서
타겟팅, 네.
이거 몇 번 맞으면요. 집안이 망해요. 엄청 비싸게 맞아요. 이게 부가가치가 엄청 높은 사업이에요. 음. Did you understand, 쉐바? 이게 엄청 비싼 이게 엄청난 일이에요. 이거 진짜 고부가가치 사업이에요. 음. 그래서 이거 몇 번 하면은 그 영화에도 보면 그런 영화 있어. 미국의 변호사인 부부가 둘 다 동시에 암에 걸린 거예요. 그래서 치료를 미국의 의료보험은 거의 무법지대거든요. 미국의 의료보험은 서부 지들 권총 싸움 하는 거랑 똑같아요. 한마디로 중구난방에 개판은 이 5분 전에. 그래가지고 그 변호사 부부가 미국에서 부부면은 거의 하이소사이트인데 부부가 둘이 치료를 받았는데 그 사람들도 보험이 있었겠지. 받았는데 두 명이 받으니까 망했어요. 그리고 집도 없어요. 집도 팔려가지고 날아가서 이제 노숙자 순서로 전략하는 그런 거 보이죠. 그거 할 때 이런 약이 치명적이지. 요즘에 우리나라가 의료보험이 아무리 잘 됐다고 하지만 요런 거 몇 개 걸리면은 우리나라 의료보험은 버틸 수가 없어요. 그러니까 글로벌 제약회사들이 어떻게든 하나 만들어가지고 뜯어 먹으려고 노력을 많이 하는 거예요. 그 사람들은 인류를 뭐 생각하고 뭐 그런 거 전혀 없이요. 어떻게 하면 베껴 먹을까? 그 생각밖에 없이요. 얼마나 악독한데, 그 사람들이. 이번에 코로나 때도 보면 그 뭐야, 그 mRNA 저기 뭐지야. 그 어디지? mRNA 백신 만드는데.
아스트라제네카.
이번에 코로나 때 알았어요. 아스트라제네카는 정말 좋은 기업이구나. 그 기업은 좋은 기업인데 그 코로나 백신이 그렇게 실패할 줄 몰랐지. 근데 아스트라제네카는 좋은 기업이에요. 그 사람들 하는 짓을 보면 진짜 그게 아마 스위스에 있나요? 스웨덴에 있나?
네, 스위스요, 스위스.
스위스에 있으면서 보면 그 업체들이 유럽에 다 퍼져 있는데 아스트라제네카는 정말 훌륭한 기업이고 그에 비해서 미국 기업 어디야? 어디야? 기억이 안 나네. 그 Sheba, do you remember the company, the COVID-19 company making mRNA vaccine?
Pfizer? I don't know.
파이자, 모더나,
Pfizer.
파이자. 그 회사들 하는 짓 보면은 거의 나쁜 자식들이야, 그놈들. 그렇게 많은 부작용들이 나오는데 걔네들이 하는 짓 보면은 부작용에 관한 페이퍼를 찾을 수가 없어. 걔네들이 다 손을 쓰는 거예요. 우리나라 오늘 신문 보니까 코로나 백신 부작용 케이스 이제 신고받는다고 그러더라고요. 보상을 해주려고. 그렇게 많은 그게 있었는데 얘네들 안 봐도 비교온 게 나쁜 기업이라는 게 그런 부작용이 나오는 케이스 페이퍼들을 사전에 판단하지 거의 발견할 수가 없어요. 그에 비해서 아스트라제네카는 굉장히 오픈됐고 정말 위대한 기업인 것 같아요. 이번에 알았어요. 재밌더라고요. 나쁜 자식들이에요. 그 모더나하고 거기 파이자.
제가 지금
Do
그거
you
받아야
agree
된다.
about that? 제가? 네, 그래서 이게 엄청 별거 아니지만 엄청 돈 있는 사업입니다. 하나 들어서 이게 날아갑니다. 요즘에는 모든 질환에서 치료법이 없는 질환은 없잖아요. 환자가 몸이 안 되거나 돈이 없어서 포기하는 게 대부분 사람. 근데 의학은 그런 게 아니잖아. 돈이 없어서 포기하게는 만들지 말아야 되는데 의학인데 그렇게 만들고 있지. 자, 그러면 다음 주에는 누구신데요?
다음 주에는 김도경 학생이 아마 할 거예요.
네.
그, 지금, 제가 지금 쓰고 있는 논문 지금 데이터 다 나와가지고 지금 쓰고 있는데, 제가 이번 주 금요일 날 미국 가기 전에 다 써놓고, 도경 학생한테 발표하라고 지금 준비시킬 거거든요. 그 친구가 지금 그래프 만들어 놨으니까 아마 잘할 거예요. 영어로 해도 잘할 거예요.
김도경 학생? 김도경 학생. 김도경 학생. 네.
예,
김도.
예, 영어로 해도 잘할 거예요. 아마 영어로 할 거예요. 아마.
오, 그럼 난 음, 오케이. 그러면 다음 주에 뵙겠습니다. 쉐바,
네.
굿잡.
Thank you. Thank you.
응, 응.
Thank you, Sheba.
예.
Bye. See you next Tuesday.